By-Induprava Sahu. Welcome you all to our YouTube channel Sneb'S. So today we will be discussing about gene interactions. Okay, and in gene interactions we will discuss about epistasis. Okay, so our main topic of discussion epistasis. Okay, so before going to discuss gene interaction, let me tell you, if you are new to this channel or if you have not yet subscribed to our channel, then do subscribe. Press the bell icon next to the subscription button, select all so that you can get all the notifications whenever a new video comes up. Okay, hit like, share and don't forget to subscribe. Okay, so we know that gene interaction takes place, right? How? Because of the phenotypic trait. As the genes interact, that's why what? Many characters are expressed in various organisms, right? Clear? So, we have studied about Mendel's monohybrid cross, Mendel's dihybrid cross, okay, and various deviations to Mendelism, right? So, there we have studied that a gene is there, right? And a gene has the alternative forms, okay, clear? Alleles, okay. The alternative forms of a gene are known as what? Alleles, clear? So, one allele is dominant allele, right? And the other allele is the recessive allele, right? This thing we have studied in case of what? Mendel's law of inheritance, that one allele will be dominant and one allele will be recessive, okay? And these two alleles of a gene, they interact to produce a phenotypic trait, right? But in the deviation of Mendelism, what we have studied? We have studied two types of things, okay? This is the first one where one allele is dominant and the other one is recessive, okay? Another type of interactions, okay, that we have seen is that two alleles, okay, express themselves to some extent, okay, clear? And here the phenomenon that we have studied is known as what? Your partial or incomplete dominance, right? Clear? Now, another thing that we have studied is that the two alleles express themselves to same extent, okay, clear? And in this case, the phenomenon is known as what? Your codominance, right? Clear? So here what happens? In case of dominance, this phenomenon is known as what? Dominance, okay? So in dominance, incomplete dominance and codominance, what happens? The two alleles of a gene, they interact, right? Clear? So this type of genetic interaction is known as what? This type of genetic interaction is known as intra-allelic or intragenic, or we can say genetic interaction. Understood? Clear? So what is intra-allelic or intragenic or intragenetic interaction? The interaction where the two alleles of a single gene interact. Okay, clear? In that case, we see three conditions. If one is masking the effect of the other, okay, alleles, okay, one allele is masking the effect of other allele, then that phenomenon is known as dominance, right? If both are expressing themselves to some extent, okay, then that phenomenon is known as partial dominance or incomplete dominance. and if the two alleles express themselves to the same extent, then we call it as the codominance. Right? Understood? Clear? So, another type of genetic interaction is also there. Okay? And that is known as, this is one type. Okay? And another type is known as your inter-allelic or intergenic genetic interaction. Okay? Clear? So here what happens in case of inter-allelic genetic interaction? Here many genes interact. Okay? And the genes that interact are present on the non-homologous chromosomes. Okay? Two different chromosomes. Understood? Clear? So, the genes are present, the genes are different because they are present in non-homologous chromosomes. Okay? And they interact to produce a phenotypic trait. Okay? So, in case of dominance, incomplete dominance, codominance. Okay? We found the ratio what? 9:3:3:1 or 1:1 ratio. Right? But in interallelic genetic interaction, we found the ratio what? Other than 9:3:3:1 or 1:1. Okay. So, let's see. So, epistasis. Okay. So, let us first discuss something about gene. Okay. So, we know that gene is what? Gene is a chemical determiner. Right? Clear? And as a result of the interactions of many genes and their products, what happens? Phenotypic trait results. Right? Clear? And these products, they interact with the environment in various ways. Right? So, when we are saying the environment, okay, then it refers to the two types of environment. What are they? One is your ecological, or what we say, the external environment. Okay? And another one is your internal environment. Clear? So, when we are saying ecological or external environment, we generally refers to what? We generally refer to your temperature, right? Light, these things. Clear? But when we are saying the internal environment, we generally refer to what? Refer to the hormones, okay, and enzymes. Clear? So, these hormones and enzymes are ultimately controlled by, the regulation is ultimately controlled by what? Controlled by the genes. Okay? So, we know that enzymes are proteinaceous, right? So, enzymes, all enzymes are proteins, but all proteins do not act as enzyme, right? So, when these enzymes act, then what happens? Some product is formed, right? Clear? So, we all know about this term metabolism, right? So, what is this metabolism? It is a process which involves two processes. One is catabolism and other one is what? Anabolism, right? So, when we are saying catabolism, it means what? Your breaking of substances or molecules, right? It is the process of breaking of what? Your bigger molecules into smaller ones, right? But anabolism, it is a process of making of substances, right? Making of substances, making of bigger substances or bigger molecules from the smaller ones, right? And hold together, the process is known as what? Your metabolism. Okay. So, in our body, various metabolic pathways are going on, right? Like respiration, like digestion, absorption, everything, okay, comes under this metabolism, right? So this metabolic processes, okay, involves what? Various steps, right? It involves various steps where some substance is there, right? Some precursor molecule is there, and that precursor molecule get converted into various subsequent molecules and ultimately gives rise to the final product or the end product, right? So there is a precursor, clear, and in various steps, okay, we get the end product, right? Clear? And each of this step is controlled by what? Is catalyzed by what? Enzymes, right? Each step of the process, okay, this process where the precursor get converted into the end product, the steps are catalyzed by what? Enzymes, right? Various enzymes. And these enzymes are what? These enzymes are proteins, right? And the organization of proteins, okay, is dependent upon what? Dependent upon genes, okay. So we can say that the enzymes are the ultimate products of what? Genes, right? Enzymes are the ultimate products of what? Genes, clear? As genes interact, okay, a phenotypic trait is produced, right, through this thing, clear? So all this process, okay, where the precursor get converted into the end product and each step catalyzed by various enzymes, okay, is what? Is called the biosynthetic pathway, right? Biosynthetic what? Pathway, clear? So we can say that the biosynthetic pathway is ultimately controlled by what? Is ultimately controlled by genes and their products, right? So let us understand this phenomenon of epistasis, okay, in this biosynthetic pathway, okay, by taking the example of a biosynthetic pathway, okay. So now consider a biosynthetic pathway, okay, where there is a precursor, okay, and the precursor is P, clear? And there is an end product, okay, and the end product is what? C, understood? So first the precursor get converted into the product A, okay, and then it get converted into the product B, and then B get converted into C, okay. So as we are taking a simple biosynthetic pathway, okay, so here we have how many steps? 1, 2, 3, okay, we have 3 steps, okay, and we know that in a biosynthetic pathway each step is catalyzed by a specific enzyme, okay. So let us take the enzyme for this step as E1, okay, for this E2 and for this E3, okay, clear? And previously I have told you that what? The production of this enzyme is ultimately regulated by a certain specific gene, right? So let us take 3 wild type genes, okay. One is G1, G2 and G3, okay, and for wild type let us write plus, okay. So these are the dominant forms, okay, and I am writing the recessive forms in black, okay. So G1, G2 and G3, okay, this is the recessive forms of the gene, okay. So let us consider that when these genes are in dominant condition, okay, wild type condition, then what happens? Then only the production of enzyme is normal, okay, clear? and the biosynthetic pathway is taking place normally. Understood? Clear? So now, if this G3 plus, okay, becomes homozygous recessive, means what? G3 अगर ऐसा हो जाए तो, okay, then what happens? The production of this one will be hindered, right? So as a result of this, what happens? C won't be produced, right? And as G2 plus is in normal condition, okay, it is in dominant condition, wild type condition, then what happens? B is produced, okay, but it cannot get converted into C, right? And B gets what? Accumulated. Clear? So B gets accumulated and C is not being produced, okay, as this gene is faulty. Understood? Clear? Now, if G2 plus, similarly, okay, now forget this one, okay, so now if G2 plus is not working properly, okay, it is in homozygous recessive condition, okay, that means what? G2 is present, okay, G2 plus is absent. Clear? This form of gene is absent. Then what happens? This won't be produced normally, right? Clear? So if enzyme 2 is not produced normally, then what happens? Then this step cannot be completed, right? Clear? So this won't be produced, okay, and this get accumulated, right? A get accumulated and B will not be produced. Clear? Now, so this one was faulty, okay, in the example, but this was not faulty, right? Clear? This is working properly. Clear? So this one is producing what? E3, right? Clear? So it has the ability to convert B into C, but as G2 plus is not working properly, okay, it is in homozygous recessive condition, okay, E2 is not being produced, as a result B is not produced, right? And this has no effect, right? Understood? So what? This is, the middle one is what? Faulty, okay, this one is faulty, okay, this one is in proper condition. Clear? But what happens here? Here, because of this one has no effect, right? Clear? So when G2 plus is in homozygous recessive condition, okay, this gene is homozygous recessive, okay, homozygous recessive condition, okay, and this one is in dominant condition, okay, then what happens? This one is masking the effect of this one, okay, so G2 plus masks what? G3 plus, right? When in homozygous recessive condition, right? Clear? So this gene is masking this gene, the effect of this gene. Clear? So the phenomenon where one gene marks the effect of the other gene is known as what? Is known as epistasis. Understood? Clear? So what is epistasis? Epistasis is the phenomenon where one gene masks the effect of other gene. Okay? Clear? Understood? So this epistasis, this is a Greek word. Okay? Clear? And it means what? It means standing upon. Okay, clear? So here in our example, what happens? G2 plus when present in homozygous recessive condition is tending upon G3 plus. Okay, as a result of which G3 plus is not able to express its effect. Okay, clear? So here we have two terms. One is hypostatic gene, okay, and another one is your epistatic gene. Clear? We have these two gene types. Okay, so what is epistatic gene? The gene which masks the effect of the other. Okay, that type of gene is known as what? Epistatic gene. Understood? And what is this hypostatic gene? Okay, the gene whose effect is masked. Okay, clear? Understood? The gene whose effect is masked, okay, is known as hypostatic gene, and the gene which masks the effect of the other is known as what? Your epistatic gene. Understood? So in this case, what happens? G2 plus acts as the epistatic gene, right? Where G3 plus acts as what? It acts as the hypostatic gene. Clear? Because this one is masking the effect of this one. Clear? Understood? So this is your epistasis. Okay. Now, one confusion here arises. Okay. The difference between the dominance and epistasis. Okay. So the phenomenon of dominance is what? In case of dominance, what happens? Here, the two alternative forms of a gene is present, okay, which we call as alleles, right? So in case of dominance, one allele is masking the effect of the other allele, right? One allele is there, okay, which is dominant allele. The dominant allele is masking the effect of the other allele, okay. But in case of epistasis, what happens? One gene is masking the effect of the other gene. Okay, clear? So in dominance, we say allele. Okay, allele is masking the effect. Okay, but in case of epistasis, what? Gene is masking the effect. Understood? Clear? Now another thing is there also. In case of dominance, what happens? We see the ratio of 9:3:1. Right? But in case of epistasis, we will get the ratio which will be the combination of these four numbers. Okay, various combinations. Okay, that we will discuss in the next video. Okay, so this is all about epistasis. We have discussed about genetic interactions in this video, and I have told you about the intra-allelic genetic interactions and the inter-allelic genetic interactions. Okay, and how the gene works, okay, in a biosynthetic pathway, and from that biosynthetic pathway we got to know about this epistasis, right? And in epistasis we studied about the hypostatic gene and the epistatic gene, okay, and the common difference between the what? Dominance and epistasis. Okay, so this is all about this lecture about epistasis, and I hope you all have understood well. And still, if you have any doubt, then you can ask me questions through the comment section. Okay, hit like, share, and do subscribe, and don't forget to press that bell icon. Thank you.